Informatic Tools Award focused on the development of ProKino, a protein kinase oncology browser.
Thanks, Larry. Yeah, so I was going to talk a little bit about some of the tools we're developing as part of the cutting-edge informatic, tool development, because we're really funded to develop informatic tools as part of this consortium. But because I've been asked to talk about the target watch list, I thought I would instead sort of focus on the other aspect of my lab, which is to really look at some of the dark kinase functions. And I was going to illustrate in the context of the symposium how pseudokinases are really understudied targets and how they really present a valuable set of targets for the kind of approaches we've heard so far. But before I do that, I just wanted to give you a quick pitch on the tool development itself. So as Larry mentioned, much of our focus is on developing the protein kinase ontology. And so this is an integrative knowledge map that really integrates and conceptualizes a lot of the information on the structural and regulatory features of kinases in human and machine readable format. And then we're also developing these comparative kinomics approaches, which really taps into the evolutionary features of kinases and allows comparative analysis of dark kinases in the context of well-studied light kinases. So I would encourage you to kind of explore these tools that are now publicly available through these links. But for today's talk, I thought I was going to focus, since I have only 10 minutes, on a couple of targets that are related to pseudokinases. And since we haven't heard about pseudokinases so far, I thought I should probably introduce to you guys what pseudokinases are and what pseudoenzymes in general are. So here's a simple cartoon describing our view of how these pseudoenzymes came about and why they're important in the context of cellular signaling and functions. So when you have a canonical kinase or an enzyme, it's capable of performing its catalytic functions. But enzymes also have these so-called non-catalytic or protein-protein interaction functions. And so when they evolve either through gene duplication or through accumulation of new mutations, some of these mutations sometimes happen in the active site, and that results in a non-catalytic or a enzymatically dead enzyme. However, these catalytically dead enzymes nevertheless retain some of the non-catalytic functions, such as protein-protein interaction functions. So, and for a long time, these so-called catalytically dead enzymes were ignored because you couldn't really assay for them because they don't have detectable catalytic activity. But nevertheless, as I said, they maintain important non-catalytic functions. And now we're realizing that these sorts of catalytically dead enzymes are highly prevalent across the genome. In fact, many gene families encode these so-called pseudo enzyme components. Fittingly, there's a recent review to which we've contributed, really highlighting the unexplored functions of these pseudo enzymes. So I would strongly encourage you to read this recent review that highlights the functions of these pseudo enzymes. But for today's talk, I thought I'll just very quickly talk about sort of two targets that we've been directly involved in the context of kinases. So as Sean mentioned, this is the kinome tree, and out of the 534 kinases, about 50 of them are predicted pseudokinases because they lack one or more of the catalytic residues. And out of the 50, about 11 or 12 of them are dark, as described by Tudor, based on the criteria that Tudor described yesterday. And so we've been closely involved in a collaborative effort on characterizing some of these pseudokinases, particularly TRP2 pseudokinase that's involved in cancer and several neurodegenerative disorders. And more recently, in a collaborative effort trying to characterize ULK4. So I wanted to very quickly introduce these two kinases and pitch them as sort of novel targets that are worthy of high throughput drug screening or computational screening, as that we've heard so far in the ATOM symposium. So these TRP2 pseudokinases are really functioning as scaffolds. So we cannot detect catalytic activity. Nevertheless, as I mentioned, these pseudokinases can function as scaffolds by really bringing together the substrates, in this case, with ubiquitin ligases, which in turn, ubiquitinate their substrates, thereby facilitating degradation of the substrates. And so, and there's other functions of TRP2 pseudokinases which I won't go into, but based on sort of constraint-based modeling, which is really identifying the unique constraints imposed on TRP2 kinases, 3 years ago we were able to come up with the structural model of how, despite the lack of the catalytic residues, the TRP2 pseudokinases actually stabilize a unique conformation of the activation loop that allow this sort of allosteric coupling between these COP1 binding region in this N lobe of the kinase with the substrate binding region, which is in the C lobe. So here's an example of how the variation in the catalytic site is used or repurposed for a new function of allosterically coupling this ubiquitin ligase with the substrate. So, and subsequently, Pat Iyer's group kind of used the thermal stability or DSF screen-based approach to see if there are known kinase inhibitors that could potentially bind to this TRP2 pseudokinase. And using the sort of thermal shift assays, they identified several EGFR inhibitors that could either stabilize or destabilize the TRP2 pseudokinase. And in particular, the focus was on this particular EGFR type 4 inhibitor, afatinib, that was, significantly destabilizing TRP2. And Dan Folkers in Pat's lab subsequently showed that some of these EGFR destabilizing inhibitors actually contribute to degradation of TRP2 in a time-dependent manner, and that this degradation is proteasome dependent in the sense that in 16 hours with the afatinib, you have significant degradation, but you can kind of rescue that with a proteasome inhibitor, MG132. So this is all published work, but the reason I kind of highlighted this is because even though pseudokinases lack catalytic activity, they nevertheless are important drug targets, and you can potentially screen them with existing kinase inhibitors, as we've illustrated here. And so this is not a high throughput docking approach, but using sort of evolutionary constraints, identifying conserved cysteines in allosteric sites, we were able to kind of propose a model of how this EGFR inhibitor could potentially covalently bind to TRP2 through this conserved cysteine in an allosteric pocket. And so that led to a sort of a model where destabilization of the TRP2 pseudokinase by afatinib with the EGFR inhibitor could lead to ubiquitination and degradation of TRP2. So that's sort of one story. And in a couple of minutes, I wanted to talk about this other dark pseudokinase ULK4, which is, again, there's very little information on them, but almost all existing data point to an important role of these kinases in mental disorders, particularly schizophrenia. So in a recent structure that Stefan Knapp solved, we collaborated with him, and as we predicted previously bioinformatically, that some of the key catalytic residues in the ULK4 kinase are different compared to a canonical kinase. But what was really intriguing about this ULK4 is, despite lacking these key catalytic residues that are critical for ATP binding, ULK4 was still able to bind ATP. Although we don't fully understand why a pseudokinase needs to bind ATP, at least one of the hypotheses is that perhaps the ATP is functioning as a scaffold to stabilize this kinase in a unique sort of functional conformation that is critical for its non-canonical functions or protein-protein interaction functions. And so that's what we're exploring right now. So again, using these evolutionary constraints, we've identified novel allosteric sites in ULK4, which we think is critical for interacting for protein-protein interactions, as identified through a bio-ID approach that Sean just mentioned. But more importantly, I think the same allosteric—
1 minute.
Yeah, I'm almost done. You find a similar allosteric pocket in different pseudokinases as well as canonical kinases, which I think could be sort of potentially encoded through in these machine learning type approaches that we've heard from Kevin, where he described how he was encoding the geometric features of ligands, but if he could potentially encode these conformational or structural features of targets, could that lead to more potent inhibitors? That I think that's an interesting discussion to have. So So with that, I want to stop and kind of summarize that the pseudokinases are really important targets to be thinking about in terms of the development of selective inhibitors. I've just illustrated a couple of examples, but the broader take-home message is that there's really hidden sort of allosteric and regulatory sites in these kinases, these pseudokinases that are waiting to be discovered. And so with that, I'll probably stop, and I'll be happy to take questions at the end, or you could put it in your chat box. Thanks, Larry.